Assalamu alaikum girls. So the next chapter is unit number 15, that is your inheritance. Now the name of the chapter is inheritance, and in this chapter we'll be studying about the laws of genetics, the basis of genetics, the composition of the DNA, how did um, how did the study and the interest of the scientists in genetics was started, Mendel's law, all these things. So starting about the chapter. So, as we all know, that according to the human history, people were unaware of the scientific details of how do the babies they have the characteristics of their parents, and they had thought that like there would be some hereditary connection, but they never knew what is the mechanism, and they never understood. There were many questions about how they have the looks of their parents, how they physically resemble their parents, and actually, Gregor Mendel. Mendel was the person who got really interested in the genetics, and then he worked uh, like he he worked very hard to know the basis of genes and genetics. Okay, so what is the introduction of genetics? It is actually the branch of biology in which we study inheritance. Okay, so inheritance means the transmission of characteristics from parents to offspring. This is all you know from your earlier grades. This is actually revision. So these characteristics are called traits. For example, the height, the color of the eyes, the intelligence, etc. All these are inheritable traits. So actually your intelligence also belongs to your parents. Parents pass characteristics to their young ones through gene transmission. Equal numbers of chromosomes from each parent are combined during fertilization, and the chromosomes carry units of inheritance which are known as the genes. So the genes are passed on from the parents to the offsprings. Understand? Chromosomes and genes. Genes consist of DNA. So the genes they consist of DNA. They have specific instructions for the protein synthesis. To know the nature and the working of genes, we will have to study the chromosomes in detail. Okay, so what is chromosome and what are DNA? What is the difference between these two? The two chromosomes of a pair are known as homologous chromosomes. The two chromosomes of a pair. Why do we say that the chromosome exists in pair? Why? Because in human body there are 23 pairs of homologous chromosomes for a total of 46 chromosomes. We recall When we recall the process of meiosis, the two members of each chromosome separate. They separate, and each of them enters into one gamete. Remember that the meiosis is the process which is responsible uh, for the continuation of the number of genes. Why? Because the number of genes always remains same. We need how many? We need 46. So 23 pairs, and each of them they are received from a single parent. So gametes, always remember diploid and haploid. The gametes are haploid, and when two gametes meet, they form a diploid zygote. So chromosome is made up of chromatin material which is known as chromatin. It is a complex material. So chromosome is made up of chromatin, and chromatin is a complex material, and chromatin is made up of DNA and proteins. Like the histone proteins are the proteins by which the DNA is made. The DNA wraps the histone proteins and forms round structure which are known as nucleosomes. So now you know chromosome is made up of chromatin. Chromatin means DNA and proteins. And the DNA and proteins together they form nucleosomes. DNA is also present between nucleosomes. In this way, the nucleosomes and the DNA between they look like the bead on the strings. Okay, now see this is the DNA. This is the DNA, and this is the histone protein. This is the histone protein. So the red balls, like the circular structures, are the histone proteins, and these spiral structures, the helical. Now you can see the double helix of the DNA. The structure of DNA is like this. And can you see this structure here? Yes. This structure of DNA is wrapping the proteins, and these proteins are known as histones. So this is the DNA here. This is the DNA. These are the histone proteins, and together they both are known as histones. Nucleosomes. And the DNA is present in the form of the string, so it is present between the nucleosome also. So actually we can say that they exist in the form of beads in string. The fiber consists of nucleosomes, and they condense into compact forms and get the structure of. They see the structure is compact. Are they spread? Are they present, like are they scattered somewhere? No, they are arranged in a proper manner, and that manner is very compact. Compact means short structure, not a long and a lengthy and structure. See here, if you are going to if you are going to wind something, like you are making what, you are making it smaller, the structure is compact. Understand? As we all know that the Watson-Crick model of DNA was designed by James Watson and Francis Crick, and they proposed the structure of the DNA according to the fact that they have two polynucleotide strands which are interwound with each other. They are coiled around with each other, and they are forming a double helix. As you all know, the structure of double helix is this one. So if you see the structure, this is the double helix structure, and the polynucleotide strand, poly means many and nucleotides are many here. So many nucleotides, they join to form DNA. This is the Watson and Crick's model. And see, there is a, actually what happens is that there is a sugar backbone. If you see the structure closely, there is a phosphate backbone, like the phosphate sugar backbone, this pink one, and then there is hydrogen bonds which are denoted by the yellow color, and then there is nitrogenous, like the hydrogen bonds are also denoted by the yellow color, and the nitrogenous bases are also denoted with the help of yellow color, and the blue ones are the sugars. So so actually what happens is that there is a phosphate sugar backbone. This is the phosphate sugar backbone, and on the outside of the double helix, this on the outside of the double helix is the phosphate sugar backbone, and the nitrogenous bases are present in the inside. These are present in the inside, and now opposite nucleotides, they form what, pairs through the hydrogen bonds. Actually, ATNGC. So A will always get combined with T, and C with G. That is adenine, thymine, cytosine, and guanine. See these here. It has how many bonds? 1, 2, 3. So this is G and C. They have 3 bonds. Where is G and C? Guanine. This is adenine and thymine. They have 2 bonds. And this is guanine, G and C. It has 3 bonds. Understand? So they have 2 bonds between them, and G and C, they have 3 bonds between them. Understand? So the nitrogenous bases are present complementary. Always remember what does complementary means. Complementary means A will always have a bond with G. A cannot have bond with A will always have bond with T. It cannot have bond with G, and G has bond with C, and C with G. So this is how they are attached to each other. This will be like explained in detail when we'll study about the translation and transcription. So the pairing is very specific. The nitrogenous bases adenine of one nucleotide forms pair with the thymine of the opposing nucleotide, while cytosine forms pair with guanine. Adenine, thymine, cytosine, and guanine, these 4 bases are there, and 2 hydrogen bonds between adenine and thymine, while there are 3 hydrogen bonds between cytosine and guanine. I hope this is clear. Okay.